Hi, and welcome back to your video homework. Today we're going to be looking at protein synthesis, and otherwise, how are we actually going to read the book of instructions that is our DNA? Get ready, changing the slide, 3, 2, 1. So, let us do a little bit of a recap here. Remember that the genetic code is universal among all organisms. Doesn't matter if it's a plant, if it's a bacteria, if it's a human, an ape, a banana, anything like that. We're all going to have the same type of genetic code. So all cells within the organism are going to have the same DNA regardless of function. And when we say that, we mean that everything is made up of those A's, the C's, the T's, and the G's that we've been talking about. All organisms' genetic code is going to be determined by the order of the nitrogenous bases. So those A's, those T's, the C's, and the G's, depending upon the order that they are presented in, is how those genes are actually going to be read. Okay, this order is going to determine the identity and function of a protein. Changing the slide, 3, 2, 1. So when we actually talk about how one cell becomes, let's say a liver cell as opposed to a bone cell or a red blood cell, this is based off of cell differentiation. Okay, so cells are different based off of the genes that they express. All of these come from stem cells, which are the unique type of cells that can differentiate into a variety of specialized cells. I call them like the little cells of all the possibilities, stem cells. Okay, when we have cell differentiation, every cell has the whole genetic code that makes you, you, or makes a banana, and a flea. So how is it that all cells aren't actually expressing every single piece of DNA that's inside of them at all times? Well, that's due to a process called gene regulation. Okay, genes that are expressed are transcribed into what we call mRNA. Genes that are not expressed, we call those silent. They're simply not transcribed. Okay, we do this regulation process and we regulate the expression of genes so that the cells can conserve energy and provide it with only what it needs. Gene expression can be regulated during either the process of transcription or translation, okay? And this is incredibly important to understand. At any point along the process of making a protein, we can control the process. Changing the slide, 3, 2, 1. So, before we go any further, you absolutely have to understand what scientists call the central dogma or the central belief of science, and it's the fact that our DNA makes our RNA, which then makes our proteins. This is absolutely true, and most biological sciences are based off of this belief. Changing the slide, 3, 2, 1. So, before we go any further, RNA basics, okay? You know a lot about DNA, and you know a little bit about RNA, so let's remember. Remember, RNA is essentially 1/2 of a DNA strand. DNA is double helix. RNA is going to be a single helix. However, the nitrogenous bases are almost the same except for right here. You don't see thymine, you see the addition of uracil. Okay, so it's A, U, G, and C. And then, therefore, the rules of complementary base pairing in RNA, it's A to U and C to G. So the way I think about it is that you eat apples, cars always park in the garages. Okay, we have 3 specific types of RNA that we're going to focus on. We have mRNA, that stands for messenger RNA. tRNA is transcription RNA, and then we have rRNA, which is ribosomal RNA. Changing the slide, 3, 2, 1. So, here again, you've seen this before, it's just another visual reminder. DNA with our double helix, with our adenine, our thymine, our guanine, and our cytosine. And here's RNA, that's single helix. There's no matching base pairs up here. Okay, but they have cytosine, guanine, adenine, and uracil. Changing the slide, 3, 2, 1. So, when we start to go about making a protein, we first have to start with a process called transcription. Okay, if you think of this word, you are currently transcribing notes, as in you're looking at something and you're writing it down. And that's exactly what this process is. This is the first process in the central dogma. This is taking DNA and turning it into RNA. Okay, so if we're dealing with DNA, we need to remember that this is only going to occur in the nucleus because DNA can't leave the nucleus. We have a special molecule we have an enzyme here, RNA polymerase, which uses DNA as a template to create that single strand of mRNA that's complementary to DNA. And remember, mRNA is a messenger, so just think of it like the messenger is writing down the message, it's transcribing the message. mRNA is a copy then of the DNA strand. And it is possible right here that mistakes can be made, but if so, RNA polymerase has the ability to correct this mistake during this process. So the process occurs in 3 very distinct steps. First, the DNA has to unwind. You can't get to that code, you can't get to those nucleotides unless it is unwound from its double helix. At that point, RNA polymerase is going to match up to the nucleotides using DNA as a template, and then using those proper base pair rules, it's going to build that mRNA strand. And at that point, transcription's pretty much done. mRNA is going to leave the nucleus to go find a ribosome. Okay, so here's a picture of it right here, going from DNA to RNA, and then RNA is going to leave the nucleus, and we're getting ready to talk about the next part, which is called translation. Changing the slide. 3, 2, 1. So here it is again, transcription. So here's our chromosome. Remember that tightly condensed coiling of DNA. When you kind of unwind that DNA, we're going to uncoil it. We're going to unwind it. Okay, we're going to open it up. DNA polymerase runs along it, 5' to 3', building that mRNA. Okay, and then at that point, gets out of the nucleus. The whole DNA strand does not unwind. That's important to understand. It's only going to unwind and uncoil where the specific genes that need to be transcribed are located. Changing the slide. 3, 2, 1. So we've transcribed, but now we need to go from essentially the language of RNA to the language of proteins. And so when we talk about crossing languages, we're going to go through a process called translation. So this is the last half of the central dogma, RNA to proteins. This occurs in the cytoplasm of our cells on a ribosome. Remember, this whole time we say ribosomes are the site of protein synthesis. Okay, and it's this process, this translation process, that takes place there to create that protein. Okay, so this also occurs in only a few steps. mRNA is essentially read by the ribosome 3 nucleotides at a time. So we call those 3 nucleotides a codon. So translation reads mRNA a codon at a time, and based on that, we're going to look at the steps here again just a little further. Changing the slide. 3, 2, 1. So once the codon has been read, this is where we get tRNA. Okay, I like to think of this as it's transfer RNA, so it's going to transfer the appropriate amino acid to the ribosome and match it to the codon using the tRNA's anticodon. Okay, all this means is that this is the opposite form using the base pairs to the codon. Okay, so those amino acids get brought to the ribosome. This is where peptide bonds are going to connect the amino acids to start making that protein. And this happens all the way down the mRNA until we have what's called a stop codon, which actually physically signals the translation to stop. So here we have another process. DNA to RNA. This part happened in the nucleus, transcription, and then at the ribosome in the cytoplasm, RNA is translated then into protein. So we can see every 3 letters right here then becomes an amino acid. Those amino acids get linked together through peptide bonds, and we have our protein forming. Changing the slide. 3, 2, 1. This looks complicated. Stick with me. Okay, so this large shape right here, we have the ribosome. Okay, so the ribosome's technically made up of a large subunit, this lighter green, yellowish color, and this small subunit, the darker color. This strand right here is our mRNA that gets kind of fed through like tape, and the ribosome reads it 3 nucleotides at a time, so a codon at a time. The tRNAs are these globs right here. They come along and dock onto the mRNA through their anticodon, and they're carrying the appropriate amino acid. And here it's where the ribosome physically forces that peptide bond, and it kind of starts kicking out the newly formed protein. Changing the slide. 3, 2, 1. Here it is again. You're going to see a lot of these different types of diagrams. So if this is our whole cell, this is our nucleus, our DNA gets transcribed into RNA in the nucleus. mRNA comes out of the nucleus to the cytoplasm. It finds a ribosome. This is where translation happens. So our mRNA is read, again, a codon at a time. These tRNAs come, bringing the amino acids where they're joined together to make that protein. Changing the slide. 3, 2, 1. So this sounds great. It sounds too perfect to be true, and it's pretty much perfect, except sometimes mistakes can be made. Okay? So when mistakes are made, again, we call those mutations. So anytime something is changed in our genetic code, we call it a mutation. And the reason why this is important is once your code is misspelled, it's going to result in one or more of the amino acids being changed to make your proteins, or in that case, to not make the appropriate protein. So we have some types of mistakes that can be called silent mutations. And when we have silent mutations, this actually means that yes, a change was made, but there actually wasn't an effect on the phenotype of the individual, okay? If it occurred in an unexpressed portion of the chromosome, a portion that we're not currently reading, okay? If there is an effect on the phenotype of an individual, it's going to occur in the expressed area of the chromosome, or the portion of the genes that are currently being read. Changing the slide. 3, 2, 1. So, with DNA, we can have what are called point mutations. These are small changes in the DNA sequence, and it can occur at essentially a single point. So, substitution, we switch out one nucleotide for another. If it was an A, maybe it gets switched out for a G. If we have an insertion type mutation, this actually means that we physically put in an additional nucleotide. So if it read ATG, maybe we have now put in a C between the A and the T, and it's now ACTG. Okay, this can have big consequences. We have a deletion mutation, which means that we just remove one. Okay, so if it was ATG and we remove the T, now you just have AG. Okay. If a mutation occurs in the gametes, it can be inherited by the offspring. But if these point mutations occur in the autosomal, the body cells essentially, across somebody's life, it can't be inherited by their children. Changing the slide. 3, 2, 1. So what can we do with this? Why is it good that we know all of this stuff? Well, this is where the field of genetic modification or engineering comes in. Okay, this is where we can directly manipulate an organism's genome. We use this in animal farming. We've used this to improve crops, the production of drugs and vaccines, criminal investigations, and paternity testing. All of these things have had or are based off of being able to get into a genome and kind of splice it apart based off of genes. Okay. Molecular cloning is a technique used to express a gene from one species of an organism inside a different organism. And one of the best cases we have of this is the production of insulin. We managed to take that gene, put it into an organism that can make it really fast, and that's how we get commercial grade insulin. Changing the slide. 3, 2, 1. So another process where we can kind of see essentially the fingerprint of somebody's DNA is called gel electrophoresis. Okay, it's the visual way that we can see the unique DNA profile of people. We use this in criminal investigations and paternity testings. We actually have a way of breaking the DNA up into different sizes based on the genes that are present, and we run it through a field, and depending upon how those little bands separate, these are all little bands of segments of DNA, we can match it from one person to another, because nobody's going to have the exact same type of DNA fingerprint except for identical twins. Changing the slide. 3, 2, 1. That's it. You managed to make it through. Here are my bibliography links for all of the images. Hope you've taken great notes. Come to class with your questions and get ready to rock and roll. What did one sister chromatid say to the other? "Stop copying me!"